Hello Mindsetters, welcome to Learn Extra Live Exam School. Today we are doing life science with Aslam. How are you, Aslam?
Great. How are you doing, Kat?
I'm great, thank you. Please tell us what we'll be covering today.
Today we're taking paper 1 and we're dissecting it to do the 60% that's very important.
Okay.
And the next segment we'll do the 40%. The 60% deals with nucleic acids, that's DNA, RNA, protein synthesis, genetics, meiosis and biotechnology.
Wow, that sounds like a lot, eh?
That's
Thank
great.
you, Aslam. So you guys know that your exam is on Friday. If you don't know by now, then I don't know what you're doing, but you gotta know that you have to study, guys. Study hard and study well, because today we're going through all that Aslam told you to go through. Keep guys, I can't stress this enough. Keep posting questions on the page, guys. Aslam is here to answer everything that you need to answer, because it's so important that you guys actually get to cover everything that you need. Remember, if you don't know, our Facebook page is facebook.com/learnextra, and our Twitter handle is @learnextra. Today as well, we have a competition. For this, PC Training has given us a tablet computer to give to the lucky winner of the Unlock the Level competition. By now, you guys know, pinned at the top of our page is how many likes we need to reach. How you do this is that you share this post with all of your friends on Facebook and tell them to like the Mindset Learn Extra page, guys. Right now, we are sitting on 24,730 likes. We need to get to 24,897 likes, guys. And all you guys who share the most, we will be putting your names into a draw, and the lucky winner will win this tablet. I'm so excited to be here right now, guys. So keep liking the page, because I really, really want to give away this tablet in this show right now. So make sure that you keep posting and you keep liking. We're going to go for a quick ad break before we continue with Aslam. But keep you glued to the seats. Don't take your seats away. See you right after the break. Welcome back, Mindsetters. If you have just joined us, we are doing life science, and today we're revising paper 1. All of you have been saying, thank you so much. We really need this show because we really need to pass. Of course you need to pass, and we're going to guarantee that you pass extra well. So keep focusing, guys, and keep posting questions, queries, comments, anything that you need to post on the page. And let me know how your exams have been up to this point, and whether you're writing, and if you're really excited to write, if you're really nervous, or you're just like shaking so much that you can't even post even then. Over to you, Aslam.
Thank you so much. Guys, you've got a great team here, so you're going to be the guys that are going to give us the number that we need for the likes. We are looking at the first 60% of your paper 1. And remember, guys, if you're writing version 1, then you will write in paper 1 DNA, RNA, protein synthesis, genetics, meiosis, biotechnology, and evolution. If you are writing version 2, you will do all of what I said, what we are doing today, plus you won't do evolution. You will have reproduction in your paper. Evolution would go to paper 2 for version 2. Okay, let's go straight into the questions. We're starting with multiple choice questions just to get the juices flowing, and at the same time to teach us the tricks of elimination. How do we eliminate in a multiple choice question? Question deals with DNA. They say DNA can be extracted from cells in the lab using household detergents. The steps which follow are not in the correct order. They're giving you the steps that we need to use, but they're not in the correct order. They're saying there, ethanol is added to make the DNA visible. Ethanol to make the DNA visible. Cells are broken down by mechanical or chemical methods. And lastly, they're saying meat tenderizer is added to break down some of the proteins bound to the nucleic acids. So we can release the nucleic acids so we can see them on the surface of the solution. The correct order of the steps is, and here, people, you must understand that before you actually go into the chemical processes, you've got to break down the cells mechanically. So obviously, the one that says number 2 must be first, because we must deal with the mechanical, physical breaking down the cells first, making it smaller. We're going to cut it. We're not going to take the whole plant cell and throw it in there, or what, the tissue. We're going to cut parts of the tissue and we're going to put it into the solution, and then we will follow. So we look at our alternatives here. Wherever 2 is not first, has to be struck off. So that's out, that's out, that's out. It leaves you with C. Now, it's not always this easy in a multiple choice question. Sometimes most times you will be left with 2 alternatives from which you will have to choose one. In this case, it was easy because 2 was only given in one of the alternatives, so your answer is C. You're going to write C next to 1.1.1, or in this case 1.1.4 in your answer book. The next question. After DNA replication, the word replication gives you the clue to this answer. Chromosomes appear as a chromatid, double-stranded, single-stranded, as single-stranded daughter chromosomes. Now, a replication tells you that something is being duplicated, a copy is being made. So if a copy is being made, then there must be 2. So anything that has single had to be struck off. Single-stranded, out there. So we're left with A and B. A chromatid, if you know your work, chromosome is made up of 2 chromatids. So each strand here is a chromatid. So that's also single-stranded. So chromatid is out. Your answer is B, and you will write B in your answer book next to 1.1.5 in this case, because after replication, the one strand is making a copy of itself, so there'll be 2 strands, obviously. Next question, a little bit of biotechnology in it, as well as your content on blood groups. A pregnant woman was told by a genetic counselor that her baby had equal chances of having blood type A or AB only. So whatever baby, whenever she has a baby, her babies or baby will have a 50% chance, equal chances, of being A or AB only. So that means this baby cannot be O, and this baby cannot be blood group B. Can only be A or AB, and that too, equal chances. Now here, if you know your Punnett square story, it becomes easy. But before you even go that way, you look at this situation here, and you look at D. Because this is AB, and here we have A and O, we know B is dominant to O. So when this B and that O get together, you're going to get blood group B. So that one there, we said can't be, so it's out already. This one here, this one is homozygous for B. So again, here's an O on this side. That B with that O will give you B again. So that one is out. So we're left with these 2 here. Here you can see clearly, again, if you take that one and that one, you're going to get B. And that one, you're going to get B still. So you're left with this one here. Your answer is A. Now let's look at it on a Punnett square to see. Now you're checking whether your answer is correct or not. How do we do that? Simple Punnett square. We draw the Punnett square. Whatever is on the left, IA, this is blood group A, homozygous. And on the right, whatever is on the right, put it on top. IB, that is B, blood group B, heterozygous. Now let's combine them. This one would give you IA, IB. This one would give you IA, I. That one would give you IA, IB. That one would give you IA, I. So there's 2 that are blood group A, 2 that could be blood group AB. Remember, that doesn't mean that your first child will be AB, the second one. This means every time fertilization takes place, these are the probabilities of the blood groups of the children. Every time. You can end up only with AB, but the chances are that you have 50% is to 50, or we can break that into 1:1. And that's your equal chance there to get that answer. Notice, guys, by the way, just by doing this multiple choice question, we've also worked out a cross between a woman with blood group A and a man with blood group B to give us the possibilities of the children that they may have. And you'll notice when we did this, the way we work it out. Remember, I haven't done the whole format. I only looked at the Punnett square just to check our answer because this was a multiple choice. But you'll notice the way we work out the Punnett square is the same whether we're working with color of eyes, whether we are working about the sex, whether it's going to be a male or a female, whether it's a sex-linked disease, whether it's an ordinary disease, whether it's fur color, etc. The way we work the Punnett square is the same. The symbols we use may change. Whether we use capital and small may change, but the way we work it out is the same. And we are going to look at genetics later, so we'll come back to that when we get there. But the main thing is focus on what we have here, that no matter whether you're working with blood groups, whether you are working with color blindness for that matter, or any other sex-linked disease, an ordinary disease like albinism, or any other condition or color or characteristic, the Punnett square, the final answer, is worked out the same way. And the format is the same, which we will see later. Good. Now we have a question that is adapted from IEB 2010 November. Nice question. They're saying the following is a photograph of a student's model. Learners have made a model to show the composition of DNA. The model is made from different color sweets called Jelly-tots. And they use wooden sticks as well. And here's the model. Okay? So they're giving you a key on the side. The white dots there, adenine, 2 of them, 2 Jelly-tots put together, white ones. Give you adenine. The orange ones, single, cytosine, 2 orange ones here without the little fluffs there. In this case here, guanine, and then the thymine, the pinkish one, and the red one, phosphate, and the bluish, purplish one, a deoxyribose sugar. Katlejo, I see you.
Sure, I feel like I want to eat
the
them.
Jelly-tots.
Yes,
No,
that's
no,
what
no.
I want to do.
We're not going to allow that. We can't take any of these out from here. Now it's going to spoil our model.
I know, right?
No chance of that today, Katlejo. I'll buy you one later, don't worry.
Thank you, thank
okay.
you.
They say what three molecules in the key could make up one nucleotide? There's the key provided here. Of this key, which 3 molecules will make up one nucleotide? Now, let's understand the concept nucleotide first. A nucleotide is the basic unit of a nucleic acid. You need 3 components in that nucleotide to be complete. And from here, what would we need? We'd need the phosphate, we'd need the deoxyribose sugar, and any one of these 4, any nitrogenous base together. So phosphate, deoxyribose sugar plus a nitrogenous base would give us what we need here to make it up. So we need a phosphate, we need a deoxy. Remember, we are dealing with DNA, so you must specify deoxyribose sugar, not just sugar, and any nitrogenous base. That means it could be adenine, it could be thymine, it could be guanine, and it could be cytosine, any one of them. So we'll have a phosphate, a sugar, and adenine, for example. That is one nucleotide. This is an adenine nucleotide. Or we can have phosphate, sugar, and guanine. That's a guanine nucleotide. Remember, you cannot write it like that because they ask you a question, what 3 molecules? And you're naming those 3 molecules here. Good? Let's move on. Consider a list of characteristics below. They're giving you a list. List the letters A to D in your answer book. And next to each, write yes if the characteristic seems true from the model. That means you can see it in this model. Or no if it does not seem true from the model. We're not talking of your knowledge of DNA now. We're talking about this model. In other words, in other questions, they could say visible similarities, visible differences, visible characteristics. So they're asking you from this model without using the word visible now. They're saying from the model. Same thing. All right? They want to know that if DNA is divided into genes, can we see it in this model? Definitely not. So the answer is no. Adenine always joins with thymine. Let's go back. Adenine is this guy. Thymine is this guy here, the pinkish one. There's adenine. Is that thymine? Yes. There's adenine. Is that thymine? Yes. Any more on this thing? Nothing. So obviously it holds true in this case. So the answer is yes. Guanine is larger than cytosine. We go to our key. There's guanine. There's cytosine. They're using 2 Jelly-tots for that and 1 for cytosine. So obviously, if your little baby brother at home would be able to say yes there. And lastly, they're saying DNA is a flat ladder-like model. Now remember, it's very easy to confuse this because in your knowledge, you know DNA is a double helix, spiral. But the question said according to this model. And if it says, does it look like a flat ladder-like structure in this case? Yes, definitely. So our answer there too is yes. Can you see how you needed to apply your knowledge of DNA using a simple model that the learners, yourself, your own buddies could have made? The teacher took a photo of that model, and from there, get a whole question on DNA. And we can go further, say, explain how this model or this structure that we have studied makes a copy of itself. Then you'd have to explain DNA replication. Now that's content, moving away from your application to the actual content. But if you got this part wrong and you didn't know what you're talking about, then you would have been messed up with the next part as well. How might using only Jelly-tots lead to an incorrect understanding of the DNA? If we use the Jelly-tots only, how will we confuse the understanding of the structure of DNA? From your chemistry, your basic chemistry, you would know that the phosphate is different from a sugar, deoxyribose sugar, and that is different from the nitrogenous bases. In this model, they all are made up of Jelly-tots, so they all take the same structure. So we are not showing the difference in the different structures. Also, well, we're only using Jelly-tots in that case, or you could have said this differently. The model with Jelly-tots shows all molecules to be the same. Apart from the color, the shape, etc., would be the same. So therefore, that is going to give a skewed understanding of this. And remember, when you want to compare DNA and RNA especially, it's nice to show that the deoxyribose sugar and the ribose sugar structures are different. So you need to work around that to do that. In what 2 ways, besides nitrogenous base pairing, is the arrangement of the molecules in the model correct? In what 2 ways, beside nitrogenous base pairing? In other words, that adenine only combines with thymine and guanine only combines with cytosine. Apart from these 2 things, in this model, what other 2 things is true of DNA? First of all, if you look at the sugar and the phosphate, the deoxyribose sugar and the phosphate, they form the sides of the ladder. And this is true of our model as well. So the deoxyribose sugar plus the phosphate form the sides of the molecule. That's 1. 2, you'll notice in the model, deoxyribose sugar is the purple one. There's a purple one attached to a nitrogen. There's another one attached to a nitrogen. Purple nitrogen. Purple nitrogen base. Purple nitrogenous base. So it also shows that the nitrogenous base is combined, or rather is linked to the sugar and not to the phosphate. So that's our second reason. The nitrogenous bases are attached to the deoxyribose sugar. Are we okay with that? That will cover us with the first question on DNA.
Yes, we can take a break now if you're ready.
I'm ready for a break.
Let's
Katlego.
take a break, guys. During this break, I hope you have seen up on the page already that there are so many likes that are going on. Keep liking, guys. Call all your friends, your family, your parents, your aunts, your uncles, everyone, all the mindsetters in the world. Everyone must like the page, guys, because we need to unlock that level. See you right after the break. Welcome back, Mindsetters. I want to quickly remind you guys about Curio. Although you don't need a reminder because many of you are enjoying it so much, use it as an assignment or as a mock test for your life science this week, guys. It will really help you, and you can stand a chance to win some awesome prizes at the end of the week. So make sure that you join us on Curio. If you haven't seen yet, the Curio codes for the week are up. The paper 1 for grade 11 is LS5, and the one for grade 12 is LS6. These are the codes for Curio on Mixit, guys. Get your teeth into it, guys. It really will help you. And I know many of you are enjoying it already, so help a friend. If they perhaps haven't joined you yet, help a friend, let them get on it, and just share your knowledge with each other as well on the Facebook page, because it will really pay off, guys. In that exam on Friday, when you think about how you helped someone else and how well you know that question, you'll be so proud of yourself. So keep doing that. Over to you, Aslam.
Thank you, Katlego. Let's go to 215 to round up this question. What two substances would need to be added to the key if it had to include components of RNA as well as DNA? Let's go back. It gave you this whole story. Adenine, cytosine, guanine, thymine, phosphate, deoxyribose sugar. So obviously, if we are talking about RNA, we'll have to add ribose sugar, and we'd had to add the nitrogenous base uracil in our key. So that's our answer there, would be ribose sugar and uracil. We had to add to this to do that. Katlego, you had a question.
Yes, I did. Let me get it for you. This question is by Nkosazana. She asks, please do a question on how to determine the percentage of a base, and that it really confuses her.
Okay, Nkosazana, let's see. Let's go back to our model. To understand this, let's go back to our model. Let's look at the codes that we have there. Adenine, there's adenine, that is adenine. I'm going to write adenine there, and that would be thymine, and that is cytosine, and that's guanine. Okay? Let's look at this molecule. We have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 nitrogenous bases. Of these 10 nitrogenous bases, 1, 2 are adenine, 2 out of 10. Okay? How many thymine are there? There's 1, and there's another one. So there's 2 out of 10. How many cytosines are there? There's 1, 2, 3. 3 out of 10. And how many guanines are there? 1, 2, 3. So when they're testing you the percentages of the nitrogenous bases, all they're really testing is to see whether you understand that in DNA, for every adenine, there must be a thymine. So if we got 10%, if we were talking about percentages now, if we had 10% adenine, then we would have to have 10% thymine, because for every adenine there's a thymine. So if there's 10% adenine, there must be 10% of thymine. So if that was our case, 10 adenine, 10% thymine. Now, they would give you only adenine, for example. Now you need to work out the rest. So adenine and thymine, 10% each, would give you 20%, because there's equal amount. Out of 20% means 20 out of 100. So 100 minus 20, 100 minus 20 is equal to 80. This goes for cytosine and guanine together. But we know that for every cytosine, there is a guanine. Cute girl, remember? Cute girl. For cytosine, combines with guanine. So obviously, that would have to divide it by 2, so there will be 40% of cytosine and 40% of guanine. It's easy as that. The same way here, we ask you, there are 3 guanines in this particular molecule. Altogether, there are 10 nitrogenous bases. There are 3 guanine. We only told you that. So in other words, you must pick it up from your mind that if there's 3 guanines, there must be 3 cytosines. 3 plus 3 is 6. We're left with 4. 4 divided by 2, because the amount of adenine is equal to the amount of thymine. 4 divided by 2 is 2. And there's it. 3 plus 3 is 6. 2 plus 2 is 4. And that is how you answer that particular question. The key is, whatever adenine is, thymine must be the same. Or whatever guanine is, cytosine must be the same. I'm hoping, Susanna, that answers your question and takes away that confusion that you had. And from now on, when you see that question, you tell the examiner, thank you very much, I know how to deal with this question in future. We move on now to a little bit biotechnology. The flow diagram shows the first 5 steps in therapeutic cloning. Therapeutic cloning is used to grow replacement tissue for diseased or damaged cells using a patient's own DNA. So they're taking the person's own DNA and they're manufacturing from that good cells to replace the damaged cells that are causing whatever problem there is in the body. So always with this, you need to read that first to understand what it means. Then you need to look at the picture shown, the source in this case. They're giving you steps. Step 1, the nucleus is removed from a healthy somatic cell of a person with the disease. There's the nucleus, we take it out. Second, the old nucleus is removed from the egg and a new adult cell nucleus is inserted inside there. Thirdly, resulting clone cells stimulated with electricity to start cell division. It's giving a boost. It's giving a cue to say start dividing. 4, stem cells are removed from the inner cell mass of the cloned embryo. We take out the stem cells from there. And 5, the stem cells carrying the patient's DNA is grown into specialized cells in a laboratory. It's cultured. In other words, it is cultured in a lab, and then these cultured cells are replaced into the body to then replace all the cells that are damaged. This is what stem cell therapy is all about in a nutshell. Remember, you do not need to go into very much detail about this, but the question has given you the information here, and let's see what type of questions they ask. First of all, what is meant by the term clone? What is a clone? A clone is an exact copy of a cell or a tissue or an organ or an entire organism. In other words, the clone would have exactly the same material as the original cell from which it was copied. And that would include the DNA of this organism or the cell. So that's what a clone is. Second question, how many chromosomes will the nucleus in step 1 contain? Let's go back to step 1. Step 1, and I circle that for you. Healthy somatic cell. Somatic cell is an ordinary body cell, and it's a human being we're talking about. Therefore, the answer can only be 46. And in this case, you cannot say diploid, because they did not ask for the ploidy. They asked how many chromosomes. You had to give a number, and that diploid number in humans is 46. In other organisms, it would be different, and then they would have to give you that. But remember, the one on the human, you need to know that. They don't have to give it to you in the text. You need to know that human beings, ordinary body cells, have 46 chromosomes, and the sex cells have 23 chromosomes. Carrying on with the same question, write down the number or numbers of the procedures below which have been used in the above process. Now for the DBE exams, you do not have to know all of these terminology, whereas in the IEB, they need to know the terminology as well. But the answer in itself was a giveaway, because if we look at what we were doing, we were taking a somatic cell from a person and transferring it, and we go back to these alternatives. The first one, obviously, you know that we are not talking about fertilization here. Number 3, definitely a no-no. Secondly, we're not cutting the DNA and reinserting DNA like we did for insulin and other issues. So recombinant DNA story is also gone. We're not genetically modifying this organism. We're not making it any different. We are taking a cell, we're making a copy of that cell. So genetic modification will also go away, and so will that one go away. And your answer is somatic cell nuclear transfer, because we are transferring the nucleus from a somatic cell. And that is the answer for that particular question. In what female reproductive structure does an embryo first normally exist? Remember, for our the normal DBE, the section, although it's here, it would be tested more in paper 2 because it falls under reproduction, but we can talk about it here. Once you've done both, we can ask you that question. In what female structure? And that would be the fallopian tube. Why? Because fertilization takes place in the fallopian tube. So the embryo, they want to know, does an embryo first normally exist? Where does it first normally? Diploid zygote, sperm fuses with the egg. That diploid zygote undergoes mitotic division to form the embryo. Okay? 315. Why might the following steps be considered unethical? Step 2 and step 4. Step 2, we're taking out a nucleus from a cell and we're transferring it to another cell. Now, if we're looking at unethical here, we could also feel this under what we would say arguments against cloning. So they want to know unethical here. Obviously, the best answer here would be normally would say that we are interfering with nature. Or we could use a famous one, we are playing God. So you've got a religious objection to that unethical issue in terms of religion. The second one, number 4, we're removing from a cloned embryo, we're taking out some cells there. So we are interfering with a living being. So that is also considered by some as unethical in that case. Remember, on the cloning issue, it's a topical issue. You're going to get different sides of the fence. You are either going to support cloning or you can be against cloning. In an exam sometimes, they even ask you for your opinion. Remember, in those questions, give your opinion. You either, I'm for it, and why? Or I'm against it and why. Remember the first part, I'm for it or I'm against it. You're going to get 1 mark anyway, because there's no right answer there. They ask for an opinion, it's your opinion. The second part, you will only get the marks if your argument relates to what your stand was. You cannot say, I agree with cloning, and then give these arguments here, because these are from those that don't agree with cloning. So it must tell you with each other. Good, we're moving on to a question on protein synthesis. The diagrammatic from KZN September 2012. Representations show the role played by 2 nucleic acids during protein synthesis in the human body. Study the diagram and table provided to answer the question. Very famous type of question. They're giving you the process firstly, and they're giving you a table here. They give you the name of the amino acid in one side, and the abbreviation in the second column. And in the third column, the codon on the mRNA that codes for these amino acids. So in other words, ACG codes for the amino acid serine, and GUA codes for methionine. They are different amino acids. Remember, guys, you do not have to know the names of these amino acids off by heart. If this question is asked, it must always be accompanied with a table to give you this, and you need to select. So the questions that are asked are linked to selecting from this table. Name the process taking place at A and B. Go back to that diagram. There's A here. Look at what's happening here. There's some amino acids here, and they're coming and they're joining with this guy here in this area. And B is here, and what's this boundary here, which we can label already as the nuclear membrane. And what's happening here? DNA is unwinding, as you can see, and something is forming on there. So they want to know what's happening at A and what's happening at B. Obviously, this is happening in the cytoplasm, so this is translation, and B would be transcription. It's very tempting to give A as transcription and B as translation because that's the order in which they normally take place. But notice they asked you specifically where A was and where B was. So you needed to know what is happening in each of those things. And guys, in further questions, they can even ask you to explain these two processes. So you must be able to explain where translation is taking place in the nucleus, where DNA unwinds and forms a template on which messenger RNA is formed, and the messenger RNA leaves and goes to the ribosome. And the significance of DNA is that DNA is giving the code via messenger RNA because DNA cannot leave the nucleus in this case. And transcription, obviously, the transfer RNA collects amino acids and brings them to the messenger RNA and joins according to the codon given by the messenger RNA with its anticodon on the transfer RNA. It simply brings the amino acids together. They join. They form dipeptide, tripeptide, polypeptide, and proteins by dehydration synthesis, and so on. You need to know those processes. You need to articulate them as well. Here, they're not asking for that, though. Give the base sequence of the DNA that codes for tryptophan. Obviously, we need to go back to our table. There's tryptophan there. And the messenger RNA is CGU. The messenger RNA is CGU. So the DNA that the messenger RNA came from must be the opposite of this. So there where the C is would be G. Where the G is, it would be C. And U is in the place of T, and normally T combines with A. So your answer is GCA for the first one. The messenger RNA that codes for histidine, messenger RNA that codes for histidine, that's a simple reading exercise. Here's histidine, and what is the messenger RNA? CAC. Straightforward. You didn't have to be a rocket scientist for that one. You have to read from the table. Transfer RNA that codes for serine. Again, back to the table. Serine, there's serine there. And the messenger RNA is ACG. Let's write that down there. Messenger RNA is ACG. Transfer RNA, this is the codon. The transfer RNA gives the anticodon, the other side of this. So it must be complementary to this one. So where A is, remember this is RNA, so that will become U, C, G, and G, C. A, U, C, G, G, C. And that is what you need to understand in this type of question. Notice you do not have to remember this thing. What are they testing you from? They're simply testing whether you know how the nitrogenous bases combine, one. Two, that you know that in RNA there's no thymine and there's uracil. You know that messenger RNA is the codon and transfer RNA is the anticodon. So therefore, messenger RNA can be seen as the patty in the burger. The rolls on either side are the same. Only the DNA and RNA, they'll have the same sequence, the transfer RNA that is. But wherever there's a T on the DNA, you must replace that with a U on the transfer RNA. But the one in the middle, the patty, is opposite to both. And that's what we showed in this question. Tabulate two differences between molecules X and Y. Again, we need to go back to the diagram. There's a diagram. Molecule X is that guy, DNA. And molecule Y is this guy, transfer RNA. So basically what they want is the difference between DNA and RNA. Obviously, you start with double strand and single strand. And if it's a double strand, that means hydrogen bonds exist between nitrogenous bases, no hydrogen bonds. We spoke about something earlier, and that was that the DNA has deoxyribose sugar, whereas RNA has ribose sugar. And we can add to that deoxyribose sugar, yes. These guys have adenine, guanine, cytosine, and thymine, DNA, and whereas transfer RNA or any RNA will have adenine, guanine, cytosine, and uracil. So there we've given you 4 differences. You only needed 3. Explain the effect, one question left on this section. Explain the effect of the protein if G is replaced with C at point Z. There's point Z. What they're saying is we take this G out and we put C in its place. So this thing is going to read CUA now. Normally GUA was coding for methionine. So when we change that code, when we change that code, if the new codon codes for the same amino acids, there are instances where different messenger RNA codons code for the same amino acids. If that is the case, then there's no problem. However, if it codes for another amino acid, then the amino acid sequence would change, and this would change the protein as well. Sometimes this is not a very serious change, because this is what we would call a point mutation. We're taking one nitrogenous base, we're replacing it with another one. Simple, point mutation. So sometimes it may not be serious. However, sometimes even changing one nitrogenous base, like in the case of hemoglobin, when the one nitrogenous thymine is taken out and adenine takes its place in one particular codon, it changes the amino acid, and this changes the coding for hemoglobin drastically, such that the hemoglobin that forms is not the same shape and efficient or the same efficiency as normal hemoglobin, and the person suffers from sickle cell anemia. This could be fatal in some cases. So that can be serious problems.
You ready for a break?
I'm ready for a break.
Okay. So guys, let's go for a really, really quick break. During this break, remember, there's still Curio, there's still your likes, so you have a busy time during the break, and you can still ask questions on the page, guys. We're noting all your questions down. There'll still be shows, so don't worry if you don't get through everything that you want us to get through today. Don't worry, just stay in those seats, guys. See you right after the break. Welcome back, Mindsetters. What awesome questions we've been receiving on the page today. You guys really, really do want to do well, and we see that. Don't worry, we have you, and we've got your back. Don't you dare worry. Aslam, we have a question by Mahlodi.
Yes.
She asks, can the process of protein synthesis be explained, along with the difference between transcription and translation?
Okay, we take that question, and then we'll take others after.
Okay, sweet.
Okay, good. Mahlodi.
Matlodi. And Lerato also asked, quite a few learners have asked this
Quite
question.
a few of you are asking the question on protein synthesis, so we would take the time to go back and explain these things. I've done it very quickly earlier, but I feel that with the diagram, this particular diagram is actually showing the process. So I'm going to explain and use notes next to these diagrams so we can understand the difference. But first and foremost, guys, the language issue here. Transcription, translation. Transcription means to transcribe, to make a copy. So understand that first. Keep that in your mind. The word transcription means to make a copy. Translate. If I start talking to you in a foreign language here, you wouldn't understand a word I spoke. So you would need somebody to translate it into a language that you would understand. And maybe somewhere else in Africa or somewhere else where somebody is watching, and if they only speak French or any other language, they wouldn't understand what I'm saying, so they will need a translator there. Or if you're watching Seven de Laan, for example, there you have those little subtitles to translate the Afrikaans, because many of you don't understand Afrikaans, but you want to watch the program. So translate is taking something in one particular form and changing it to another form. And that's all this section is about. Let's look at what's happening here. The first part is obviously transcription. First of all, you say, where does it take place? Transcription takes place in the nucleus. What is happening in this diagram? DNA is unwinding, and as it unwinds, it provides a template. A template is a mold on which mRNA is formed. In other words, mRNA, and this is the crux of this whole section here, makes a copy of DNA. But how? According to the rules of combination. If there's A on the DNA, it would be U on the messenger RNA. If there's C on the DNA, this would be a G on the messenger RNA, and so on. So that's the first part. And this messenger RNA then leaves, there's the messenger RNA there. This messenger RNA leaves the nucleus and attaches to a ribosome. There's the ribosome there. So it attaches to a ribosome. And that brings us to transcription. First of all, messenger RNA is attached to the ribosome. And when it attaches to the ribosome, such that the nitrogenous bases are exposed, the messenger RNA now is called the codon. Why? Because it is providing the code. To unlock this code, we've got other guys that are in the cytoplasm. They are known as transfer RNA. The transfer RNA molecules, they pick up amino acids. They're picking up amino acids. As you can see, there's amino acid 1, 2, 3, 4, 5. Different transfer RNAs pick up different amino acids. The transfer RNA are simply taxis. They provide the service. They're bringing the passengers to the messenger RNA. And they combine with messenger RNA. How? According to the code. There you can see what's happening. Messenger RNA's code, the codon, was GGU. And the anticodon, which is found on the transfer RNA. The anticodon, by the way, is this part here. So let me take my arrow there. GCUA. So that is how they combine. Notice that they simply combined it. That, in other words, this is the parking bay of this particular transfer RNA. It parks there. Parking number 1, parking number 2, but they're not numbered. They are lettered. And the letters indicate where they must park. And they park in that way, simply bringing the passengers next to each other. amino acid 5, amino acid 4, amino acid 3, 2, 1. They bring them together. Your taxi takes you to the school. It doesn't take you to your classroom. It simply brings you to the school. Or it takes you to the shopping center. It doesn't force you to buy at any particular shop. What you do after that is your business. So the same with the transfer RNA. They bring the amino acids together. These amino acids then, because they're close, they make bonds with each other. They combine with each other. And when they form bonds, water is released. And this is known as dehydration synthesis. And when we get two amino acids together, we call them a dipeptide. By the way, the bond in between is known as a peptide bond. When we have 3 amino acids, tripeptide, more than that, we call it a polypeptide. More than 50, we get a protein. All of these reactions in transcription and translation are controlled by enzymes. So when you're explaining transcription, you'll say reactions controlled by enzymes. When you're talking about translation, you would say the same. So you're getting a mark for that as well. So notice the step. DNA unwinds, messenger RNA forms by making a copy of the DNA according to the template provided by the DNA. This reaction is controlled by enzymes. Messenger RNA leaves and joins on the ribosome. Translation finish. Transcription starts. Messenger RNA exposes base triplets, 3 nitrogen bases. And these 3 nitrogen bases form the codon. In the meanwhile, transfer RNA picks up amino acids and brings them to the ribosome according to the code provided by the messenger RNA. The amino acids combine by dehydration synthesis to form dipeptide, tripeptide, polypeptide, and eventually proteins. Can you see there was a code by the codon? This code has been converted from just a code to a protein. So it has become something else, translated into something else. And now we have a protein. And all enzymes control all these reactions. I'm hoping that answers your question in terms of that. We move on to some genetics. We do not want to miss out on that as well. A farmer owns 2 types of cows. One type is black and the other is white. The color black, capital B, is dominant over white, small b. The farmer makes the following crosses. Homozygous, capital B, black, with homozygous white. Heterozygous black with heterozygous black. Now, just some terminology. Homozygous, when the gene and the allele are identical. We say that it is homozygous. So capital B, there must be another capital B. Small b, another small b. Heterozygous, when the gene and the allele are different. You need to know these terminologies because in a question, the terminology would instruct you what to do. It will give you a problem, and if you don't know homozygous, you don't know heterozygous, you can't answer the question. So understand that firstly, okay? The questions are straightforward here. In which cross will all the offspring in the F1 generation be heterozygous regarding color? And that is obviously in cross 1 where you got your homozygous and your homozygous, because all the offspring from here would be heterozygous. Whenever you take a homozygous dominant with the homozygous recessive in a complete dominance question, all your offspring would be heterozygous. In an incomplete dominance, also all your offspring would be heterozygous. Their phenotype would be different. And that brings us to another word, geno and pheno. Genotype is referring to these codes that we are using here, the genetic makeup of the individual. Phenotype is it black or is it white? Have you got blue eyes or brown eyes? That would tell you your phenotype. Your genotype, are you heterozygous for brown eyes or are you homozygous for brown eyes? And so on. Let's move on with the question. Which cross will produce a ratio of 1:1 with regards to homozygous, recessive, and heterozygous offspring being produced? Obviously, guys, you either need to know your work or you need to work it out. You can already see that it cannot be the first one, because in the first one, there are no homozygous. So that one is out. It can be this one, it can be that one, or it can be that. It can't be this one, because there's no capital B here. There's only small b's. So that's also out. So it can only be this one or that one. And if you know your work, you'll know that when you take two heterozygotes, you get 75%, 25% phenotype ratio. And genotype, you got 1:2:1. And they want to know homozygous and heterozygous 1:1. This 2 here is heterozygous, so it can't be that one. Your answer would be this one here. And again, how do we test if our answer is correct? We simply make a little Punnett square. You could have done it with all, but if you know your work, can you see how easy it is? You could have worked out all this way. Let's take capital B, small b, and with two small b's. Capital B, small b, capital B, small b, capital B, small b, this one would be, sorry, capital B, small b, small b, small b, capital B, small b, small b, small b, capital B, small b, small b, small b. And your ratio is 1:1. Homozygous, heterozygous. There's two of them and there's two of the other one. Straightforward, easy. But again, guys, this section you need to practice. The marks are there to take. And remember, when you're working out your problems, when they give you a full-blown problem, you must work it out in the format provided. Otherwise, you lose unnecessary marks. Still here, in which cross will approximately 75% of the offspring in the F1 generation carry the gene for black color? And I told you that already. That would be number 2. Whenever you cross two heterozygous individuals in a complete dominance, 75% of the offspring would be of the dominant color, and 25% would be the recessive color. So the answer there would be number 2. Still with this, predict the phenotype of the offspring in the F1 generation of the crossing of number 1 when codominance occurs. Number 1. Number 2, sorry. Number 2, when codominance. Now, what is codominance? Equal, both. Co-dominate, we are both dominating. When codominance occurs, there is no one color or a mixture of colors. Both colors or both characteristics will do. In this case, codominance means that we will have black and white individuals. In other words, black with white spots or white with black spots. One of the two we would have. That would be codominant. Now we are looking at number 2, and that's this one here, heterozygous. So now there, we will use the letters capital B and capital W. Capital black, B and capital W. Katlecha, what's happening?
I'm on the page.
My tongue is doing its own thing.
Oh, yeah, no, I understand. After an hour and a bit of talking, it can get a bit tiring.
It can get tongue-tied. Eh?
Can, huh?
So we've got that. There's our first one there. Capital B, capital B, capital B, capital W, capital B, capital W, and capital W, capital W. What they asked was phenotype of the offspring. Here in this case, notice the ratio of genotype is still 1:2:1. But in complete dominance, it was 75 to 25%. Now here what happens? We have 25% or 1 black, 50% or 2 black and white, and 25% white. So they wanted to know the phenotype ratio. There you go. There's the prediction given here. 25% black, 50% black and white, and 25% white. I think with that, we can go for a small break.
Okay, let's do that. So guys, we're going to go for a really, really quick break. During this break, ask those last questions, guys, those last things that you need to know just to tie up your loose ends, just so that you can really, really get a shot at acing your exam. That's what we really want for you. So keep making your comments and queries and tell me whatever you need to on the page, because I am here for you. See you right after the break. Welcome back, Mindsetters. I hope you guys are still urging all your friends and families to keep liking the page, and I hope that you're still sharing that post of how many likes we need to unlock the level. Remember, all your names, if you have shared, will be put into a box or a hat or whatever we need to put all those many names in. We'll shake it around and we will be drawing one single name so that that person can win this PC training tablet, Android, smart pad. And I hope that you guys are really keen. I know some of you are like, please just give it to me. I really, really want it. I know I also really want it. And thank you to PC Training, because this one should be mine, but I'm going to be giving it away to you guys because you guys are more awesome. Not more awesome than I am, but you're still really, really awesome. So here you go. Keep liking the page. Over to you, Aslam.
Thanks, Katleho. We're moving on. Lots of you are asking questions on two things, monohybrid crossing and pedigree. This question combines a bit of that with sex linkage. So I want you to pay careful attention, and also we've done monohybrid crosses before this. All these questions we did before this were all monohybrid. What monohybrid simply means that we're taking one characteristic at a time and crossing it. And that's all you need to know for your curriculum, especially for version 1. So here they're saying color blindness is a visual defect that occurs as a result of a recessive allele on the X chromosome. The minute they say X chromosome or they say sex linked, remember your answer has to have the symbols X and Y in it. And we'll show you just now how that works. The family tree below shows the inheritance of color blindness in one family. So here's a pedigree diagram. This is what a pedigree diagram does. It shows you the different characteristics moving from one generation to the next. Now notice in this particular diagram, there are 3 generations. The first one is always your first generation, then the second, and then the third, and you move downwards. So you label the first line as your first generation, and from then on, you're going to move downwards. Okay? Also notice that only where the line is connected to offspring, then these offspring belong to those parents. This particular guy here, Jack, he's not on this line. He is on his own line here with Jill. Jack and Jill. Wow.
Jack.
Okay. All right, Jack and Jill went up the hill, yeah? So we have them there together, but this Jack is not an offspring of Lisa and Harry because his line is not connected to them. Okay? He's married to one of their children. So those are some of the things you need to know. Usually in pedigree diagrams also, squares, or in this case rectangles, are the males, and the oval shape or circles in the exam would be the females. Also, generally, those that are affected are shaded, and those that are unaffected are blank, or in this case, with a carrier, slight shades given there as well. So those are some of the things you need to know background. Now, they go straight into the question. They say, write down the genotypes of Lisa, Harry, and Roy. First of all, let's look at Lisa, Harry, and Roy, because names can be deceiving. Lisa, female. Harry, male. Roy, male. Okay? So that's your first clue that you have to understand. Lisa is a female, so she would be XX. Harry and Roy are males, X, Y. X, Y. So that's the first thing you need to understand. The males are X, Y, and the females are XX. Now, let's go back to Lisa. There's Lisa. Notice that Lisa's circle or oval shape there has no shades at all in it in this case here. So therefore, we can assume from there that she would have the normal gene. So the gene goes on top of the superscript to the X, XB, XB. Harry is shaded. That means he's a colorblind male. So he must have the small b. Now notice the interesting phenomenon here. Where else, everywhere else in genetics, where it's not sex-linked, when you're looking at eye color, when you're looking at the size of nose, or whatever else characteristic we're looking at, we do not use X and Y, number one, and there's always 2 genes for the thing, for the characteristic. But in the sex-linked characteristics, we only have 1 on the X. The Y chromosome seems to be too small to carry any genes. So therefore, on the males, it's found on the X chromosome. And because they do not have the Y chromosome to balance it out, the minute they have this recessive gene, they would suffer from the condition. That is why it is more common among males to have these disorders than for females. Okay, so Roy now, go back to Roy. Roy is a normal male, so he would have a capital B there. Okay, good. Move on. Use a genetic diagram, and this is where our monohybrid comes in, Katlego.
Yes.
But remember, the normal monohybrid crosses would not use X and Y. But the way we set it out, the way we work it out, is still the same. All right? Use a genetic diagram to determine the possible genotypes and phenotypes of Jill and Jack's next daughter. Jill and Jack's the next daughter. Now, let's look at this diagram properly. When we have a situation like this, on your pedigree diagram, you will always do the following. Go and write down the recessive, because the recessive is guaranteed. It cannot be anything else. The dominant one is difficult because it could be carriers and you have a problem. Not all diagrams show you carriers in another color. So you have some problems with that. But now look at this. If he had a small b, where did he get the small b from? He gets the X chromosome from his mother. So this Jill had to have a small b. And because she's normal, she would be a carrier, she would be X, capital B, X small b. Can you see how you can work backwards? Very easy when you have a recessive characteristic there. This one here would be difficult because they're not showing, because Jill, could have got this X from her father. And she got a normal B from the mother. So this person actually could have been XB, XB, or XB, X small b. But in this particular case, they're giving you that slight coloration on the ones that are carriers, so this person cannot be a carrier. She's normal homozygous. But in another situation, it could be. You've got to go down or up. If you know the one on top, it's easy, or you know the one at the bottom. The minute you have this, you're going upwards that way there. This guy here would obviously get the Y from his father, and he got a normal chromosome from his mother, X capital B, Y. Jack is a normal male, so he also would be that denotation there. Now, what they want to know? They want to know Jill and Jack's next daughter. Jill is heterozygous. Jack is normal there, so we go with that. Phenotype, Jill is a normal, but she's a carrier. And Jack is normal. The genotype, Jill will be XB, X small b, whereas Jack is X capital B, Y. And this is how you set out all your genetic problems, people. Meiosis, we can put this meiosis actually there. We write meiosis here. And meiosis takes place, it spreads the gametes out. How? I say keep whatever is on the left, so it separates them. XB, X small b. And what's on the right, keep it on top. X capital B, Y. That B and that B will give you X capital B, X capital B. That X capital B with the Y would give you that situation there. X small b, X big B. Remember, the capital B and the small b can be interchanged. Doesn't matter, problem there. And the last one there, X small b, Y. The question, this fertilization, by the way, would come higher up there before that. Okay? The question asks the phenotypes and genotypes of Jill and Jack's next daughter. But because we worked out the whole problem, we can say genotype XB, 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 X capital B, X capital B, Y, X small b, Y. The phenotype here, it would be normal daughter, here, carrier. She's normal, but she's carrying it. Daughter, here, normal son, and colorblind son. But let's go to the question again. The question wants to know the possible genotypes and phenotypes of next daughter. So they have a 50% chance of normal and a 50% chance of a carrier daughter. They're only talking about the daughter. Remember, it doesn't become 25% because you're only looking at the daughters. There are two possibilities for daughters. Out of them, one is totally normal, the other one is a carrier. And that is how you set your meiosis, your genetic diagram questions. Please make sure that you follow the roster there in that sense. If Jill and Jack have another child, what is the probability of the child being colorblind? 1 out of 4, that is 25%. We have to go back to our last one. There's the one chance of the child being colorblind, only 1 out of the 4. So it's quarter, 25%. What is the genotype of this child? It would be X, B, Y. The next question would obviously ask you, what is the sex of the child? Yes, whether it's male or female, and that tells you got Y, it's a male. You notice in this problem, mainly the males are sufferers of the disease. So that is how you set out your problems in monohybrid crossing, and that is how you answer pedigree diagram question. Go for the recessive characteristics first, fill them in. In a normal problem, that would be 2 small letters. And from there, those children or the offspring get that small b from their parents, and you work backwards or you work forwards, depending what is given in the question. We have time to go through to the essay, read the statement below, and then answer the questions that follow, or the question that follows. Sex cells are formed by a type of cell division. Sex cells, or gametes, as we know them, are formed by a type of cell division. Describe how processes that occur during the formation of these sex cells results in variation in the offspring that arise from these sex cells. Now, firstly, the verb is describe. What must you describe? Whatever comes after that. How processes that occur during the formation of these sex cells result in variation of the offspring, and that's where the situation is. Now, obviously by now you know that you only studied one type of cell division in detail, and that is meiosis. So in essence, the question is asking you, how does meiosis result in variation in the sex cells? This is what you need to understand. So first and foremost in an essay question, you need to answer, or to rather analyze what is being asked. So that's the first part. Now let's look at how we would answer a question like this. Logical sequencing is important. First of all, we talk about the sex cells. They're talking about sex cells, so we must talk about them. The gametes, or sex cells, male as well as female, in other words, sperm and egg, are formed by the process of meiosis. Because they did not give you the name of the cell division, you have to specify it in your answer. So that's where you're going to get the 1 mark there. Egg cells will be formed where? In the ovary, and sperms in the testes. So you're telling them where that is being formed as well. Then you go into the whole process of how meiosis brings about a variation. And we are basically describing prophase 1. During meiosis 1, homologous chromosomes arrange themselves in pair. So we're saying that the homologous pairs are lying next to each other. Secondly, they lie close to each other to form a bivalent. That's coming from there. Crossing over of the chromosomes of the homologous chromosomes takes place. And that's an important concept, crossing over. What is crossing over? The homologous chromosomes are lying next to each other. They twist over each other. Where they meet, this point where they meet is known as the chiasma. And what happens here? Some genes from the one homolog is exchanged to the other homolog and vice versa. So groups of genes are exchanged between the maternal and paternal chromosomes of the homologous pair. What does this do? It brings about reshuffling or recombination, and this brings about variation in the sperm or the egg that is being formed. So in other words, the first thing you're talking about is when the homologous chromosomes undergo crossing over. This allows for an exchange of genes between them, and this makes the sperms or the eggs different from the original cell and from each other. Then they move on to say that the homologous chromosomes will thereafter arrange themselves in no specific order. We can talk about a random assortment of chromosomes. They are lying at the equator in no specific order, apart from that the homologous chromosomes are lying next to each other. But the one could be on the upper half and lower half, or north and south, or east and west, in any order. Every time it happens, they can be in a different order. So there's no guarantee which way the one is going to go and which way the other one is going to go. And because of that, they arrange independently, they assort themselves independently of each other, which leads to greater variation of the gametes. So the different arrangement of the homologous chromosome will result in the gametes being produced at the end having different gene combinations. So you can explain the second part, that is a random assortment of chromosomes. This means that the way they are arranged in the equator is, there's no guarantee the way they're going to be lying there. And because of this, they will separate differently. Lastly, you'll go to mutations. You'll talk about different mutations. They're talking there. One is the point mutation, and the other one is the frame shift mutation. And we have covered this over and over again. Point shift is when one gene is replaced by another one. Frame shift, when we add or we delete some of the genes, and because, or nitrogenous base, sorry, and because of that, the whole sentence changes thereafter. And because of that, it has drastic impact on the protein production thereafter. And that, guys, is how you would answer this particular essay. Kashifa had a question there.
Yes, Kashifa asked, what questions will they be likely to get on Friday?
Kashifa, you know we can't give you that type of information because we don't see that type of work yet, ever. We don't see it, and even if we did, we wouldn't be able to tell you. But we have covered many essays in almost every section, Kashifa. Go through that, and more importantly, Kashifa, and all the rest of you, remember, it's not the answer on the board that's important. It's the way we tackle this answer, the way we answer, looking for the verb, and then answering what is come, and the logical sequence with whatever we need to do. With that, guys, I hand you over to Kashifa.
Thank you, Aslam. Thank you, guys, for joining us today. Remember to keep liking the page, guys. We have about 109 likes left to unlock the level. Please do that. Remember that evolution will be right after the show tomorrow and Thursday, guys. Keep going and keep going. Learn more, learn extra, guys. Good luck. Bye-bye.